Hello everyone. Today we will explore about DNA, the blueprint of life. When we say DNA is the blueprint of life, what it means? It simply means that DNA contains the complete set of instructions needed to build and maintain an organism. Just like an architect blueprint guides the construction of a building, DNA can be found from simple bacteria to the human being. So, DNA is the life itself. In this presentation, we will explore what DNA is, its structure, and function. DNA, or deoxyribonucleic acid, is the genetic blueprint of life. But have you ever wondered what makes up this incredible molecule? DNA is composed of two key components, deoxyribose sugar and nucleic acid, which work together to store and transmit genetic information. DNA is a long molecule made up of monomers called nucleotides. DNA is the hereditary material in all living organisms. Hereditary material refers to the molecules that stores and transmit genetic information. In most living organisms, these material is DNA, deoxyribonucleic acid. It carries genetic instructions in the form of genes. Genes are specific segments of DNA that contains instructions for making proteins, for the development, functioning, growth, and reproduction of all known life forms. Discovery of DNA. DNA discovery was a journey spanning over a century, involving brilliant minds and groundbreaking experiments. Let's explore the fascinating history of how DNA was discovered. In 1869, Swiss biochemist Frederick made a groundbreaking discovery. While studying white blood cells, he isolated a substance from their nuclei, which he called nuclein. This was the first evidence of DNA, though its function remained a mystery. Finally, in 1953, James Watson and Francis Crick, with crucial data from Rosalind Franklin's X ray diffraction images, uncovered the double helix structure of DNA. This discovery explained how DNA could store and replicate genetic information with remarkable precision. Where is DNA found? DNA is everywhere in a living organism, inside the nucleus, mitochondria, and even in small circular plasmids in bacteria. It is found within the chromosomes of the cell. DNA is tightly packed within chromosomes, which are located inside the nucleus of eukaryotic cells. Chromosomes are thread like structures made up of DNA and proteins. These chromosomes store and organize genetic information, ensuring its accurate replication and transmission across generations. DNA is stored within chromosomes, ensuring genetic stability, inheritance, and evolution. These structures protect DNA while enabling its precise replication and function. Without chromosomes, the genetic code could not be efficiently managed in living organisms. Structure of DNA. Double helix model, Watson and Crick model. So, the double helix structure discovered by James Watson and Francis Crick in 1953, with crucial contributions from Rosalind Franklin, the DNA molecule is shaped like a twisted ladder, known as the double helix. And this structure allows DNA to store vast amount of information while ensuring its accurate replication. Two strands coiled around each other like a twisted ladder. The DNA double helix is right handed. What it means? It twists in a clockwise direction. This helical structure provides stability and efficient packaging within the tiny space of a cell's nucleus. Each strand is made up of nucleotides. So, as you all can see in this diagram, nucleotides are the basic structural units of DNA, and each nucleotides consist of three key components. What are these three components? First one is a nitrogenous base, which can be adenine, thymine, cytosine, or guanine. The second one is a pentose sugar, which is deoxyribose sugar in the case of DNA. A pentose sugar consists of five carbon atoms. And third one is a phosphate group. So these parts together form a nucleotide which act as the foundation of genetic material. I have already discussed about nucleotide. Each strand of DNA is made up of nucleotide. Now let's talk about backbone of DNA. The sides of the DNA ladder are made up of alternating sugar and phosphate groups, forming the sugar phosphate backbone. The sugar in DNA is called deoxyribose, which gives DNA its name deoxyribonucleic acid. The rungs of the ladder are formed by the four nitrogenous bases. These bases are adenine, thymine, cytosine, and guanine, which are held together by weak hydrogen bonds. The base pairs are held together by hydrogen bonds. Adenine pairs with thymine and have two hydrogen bonds. Cytosine pairs with guanine and have three hydrogen bonds. These weak bonds allow DNA to unzip easily during replication and protein synthesis. DNA, the blueprint of life, stores genetic information using a unique code made up of four nitrogenous bases. These bases form the rungs of the DNA ladder, allowing cells to read and replicate genetic instructions with precision. DNA contains four nitrogen bases, which are categorized into two groups. First one is purines, which are larger. Double ring structure comprises of adenine and guanine. Second one is pyrimidines, smaller single ring structures comprises of cytosine and thymine. Each of these bases plays a crucial role in storing and transmitting genetic information. What does DNA look like? The nitrogenous bases follows a strict pairing rule known as Chargaff rule. Adenine always pairs with thymine. Cytosine always pairs with guanine. These pairs are held together by hydrogen bonds. Adenine thymine pairs have two hydrogen bonds, and cytosine guanine pairs have three hydrogen bonds, which making them stronger. This complementary base pairing ensures DNA stability, accurate replication, and error-free genetic transmission. As before, we have learned about what is DNA, what its structure. Now, moving forward, we are going to explore functions of DNA. So, DNA performs various functions. Genetic information storage. As we all know, DNA stores genetic instructions that determine the traits of all living organisms. For example, the gene for eye color determines whether a person has brown, blue, or green eyes, and these instructions are encoded in the sequence of nitrogenous bases. Second function is mutation and variation. Mutation and genetic variation. What do you mean by the term mutations? Mutations are changes in the DNA sequence which can occur due to errors in replication, environmental factors, or random changes. Some mutations are harmful, some beneficial, while others have no effect. Genetic variations occurs due to mutations and recombination, driving evolution and diversity in species. Example: mutation in the hemoglobin gene cause sickle cell anemia, but the same mutation provides resistance to malaria. Third function is replication. Before a cell divides, DNA undergoes replication, ensuring that each new cell receives an exact copy of genetic material. DNA replication follows a semi conservative model. DNA replication allows organisms to grow, repair damaged tissues, and reproduce. Fourth is regulation of gene expression. DNA does not produce proteins all the time. It regulates which genes are active or inactive based on the cell's needs. For example, insulin is only produced in pancreatic cells, even though all cells contain the insulin gene. So, DNA is responsible for the regulation of gene expression. So, today we have learned about DNA, its structure and function. DNA is more than just a genetic code. It plays a crucial role in storing information, ensuring replication, allowing mutations for evolution, and regulation gene expression. These functions make DNA the foundation of life and biological diversity. Thank you for your active listening, and that's all for today. Bye bye.